When people are walking through the bush, they might come across these plants and maybe not really notice them. They might think it's a weed or something else, but if you look closely, you'll see how special they really are. I'm Laura Skates, and I'm really passionate about our Australian native plants, but I'm especially fascinated by our carnivorous plants. I was always interested in nature and the environment, but when I got to university, I started studying plants in more detail, and I learnt about all of the amazing adaptations that our native Australian plants have to all sorts of different conditions. And I learnt about carnivorous plants and how they've evolved to not only survive but thrive in nutrient-poor soils, and that we have such a huge diversity of them here. And yeah, I was just hooked on them. Right now we're in the beautiful Allison Baird Nature Reserve. It's a beautiful biodiversity hotspot right in the centre of an industrial part of Perth. We're surrounded by cars and industry and residential areas. So yeah, but it's a very beautiful, special spot. A lot of people might be surprised to find out that Western Australia is actually a global centre of carnivorous plant diversity. We've got about a third of the world's carnivorous plant species right here in WA, both here in the southwest corner and up in the northern Kimberley region. What we're looking at right here is a kind of Drossera, or a sundew. This one is a Drossera erythorhiza, or the red ink sundew. It's a tuberous carnivorous plant, a tuberous Drossera. So it has a little tuber that grows at the bottom that it'll die back to, and then it'll grow from the next season. These plants eat all sorts of bugs, basically. Any unlucky insect that happens to be walking along and gets stuck in the sticky tentacles is what the plant will end up eating. Going on a bushwalk with me, there is a lot of stopping and looking at plants and taking lots of photos. You kind of have to tune your eyes in. Once you've seen one of the Drossera with its sparkly dew, you'll start seeing them everywhere. Having a closer look at the plants really gives you a deeper appreciation of them. You can really see how they're put together and what they're doing. Oh, cool. So we have here one of the climbing Drossera, and you can see that it's climbing all the way up from down here up to here. And this one's really interesting because its leaves, basically, if you look really closely, you can see that they kind of have a shape a bit like this. And basically what they'll do is that when an insect lands, the tentacles will wrap around the prey there. But what it's also doing is that some of its sticky leaves it's using to help it climb up. So there's one here which it's holding on to this grass here, and it's just doing that all the way up. It's got these extra long sticky leaves that it's using to climb up. These plants really amaze me in how they've adapted to this environment to be able to climb up even higher into their ecosystem so they can catch even more and different kinds of prey. I've carried out a part of my PhD research here in the reserve. I was looking at a lot of different species of Drossera, so some of these climbing ones, some of the flat ones, all sorts of different species, and looking at them over time to see how their nutrition changed across the season. My research took me to a lot of different places, so I've been all over the southwest to look at carnivorous plants. I've also been up in the Kimberley region, which is another diversity hotspot for carnivorous plants. And I've also gotten to go to Germany to do all my lab work with a world-renowned stable isotope expert over there. I got to spend time there in the lab and learn these new techniques. Yeah, it was amazing. My research has been looking at basically how carnivorous are carnivorous plants. So, how much do they rely on and benefit from the prey that they capture in terms of their nutrition. So the way that I figure out how much they benefit from the prey that they're capturing is that I use stable isotope techniques. When I'm out in the field, I'll collect a small sample of leaves with a scientific licence for collection. I will dry them and bring them back to the lab. When I'm in the lab, I will grind up the dried plant leaf samples into a little powder. I measure out that powder, weigh it into these little tin capsules. The tin capsules get folded up like a little burrito and then put into the isotope ratio mass spectrometer, which measures basically the isotope ratio of the sample. We're looking specifically at nitrogen, which is an element that's really essential for plant growth and reproduction, but is also very limited in our soils here. So the plants really rely, in a way, on the insects that they capture to get the nitrogen that they would otherwise struggle to get sometimes from the soil. While I was in Europe, I also got the chance to go to the house and gardens of Charles Darwin. Going to Darwin's house was just incredible to be able to go around in the place that he lived and did all of his research, and especially his gardens and even more so his glass house where he did all of his experiments on carnivorous plants. I was surprised to learn how much work he'd done on them when I first started studying carnivorous plants, but he basically provided the first scientific evidence that these plants are actually able to capture and digest insect prey. One of the things that Darwin found was that these plants particularly respond to substances that have nitrogen in them. So that's kind of the basis of my isotope research, knowing that nitrogen is such an essential element and it's what the plants are reacting to in the prey, and that we can measure that using the stable isotope techniques. At the moment, I'm writing up all of my research to hopefully get it published and to finish my PhD. But in the future, I'd love to be able to keep working with carnivorous plants, and especially more on the lines of how do we conserve them and how do people and plants interact. That's what I'm really interested in.